AI may help rebuild medicine's shrinking antibiotic arsenal. A new system does more than search databases for molecules that already exist. It proposes changes to bacteria-fighting peptides, ranks promising versions, and sends a small set forward for laboratory testing. The early results are striking but still early. These are experimental candidates tested in laboratories and mice, not approved antibiotics or proven human cures. What did the AI design, and how much closer does it bring us to useful medicine? Antibiotic resistance is not a single futuristic disease. It is what happens when bacteria evolve ways to survive drugs that once stopped them. That can make pneumonia, bloodstream infections, wound infections, and complications after surgery harder to treat. The scientific problem has two parts. Medicine needs new antibacterial ideas, and it needs faster ways to improve the ideas that already look promising. One place researchers search is the world of antimicrobial peptides, short chains of amino acids made by many living organisms. Some can damage bacterial membranes, disrupting the protective boundary a bacterium needs to survive. Nature offers an enormous number of possible sequences. The difficulty is choosing which ones deserve the time and cost of chemical synthesis and testing. Traditional discovery often screens a library, assemble many candidates, test or predict which might work, then optimize the survivors through repeated experiments. But peptide sequence space is vastly larger than any practical library. Change one amino acid and activity might improve, disappear, or come with unwanted toxicity. Change several, and the combinations multiply rapidly. The 2026 study introduced Apex Generative Optimization, or ApexGO. It starts from an existing peptide template and searches for useful modifications rather than generating without direction. A transformer-based variational autoencoder learns a continuous mathematical representation of peptide sequences. That matters because optimization is easier in a navigable landscape than across disconnected strings of amino acid letters. Peptides form an immense molecular landscape, but only a fraction can be tested. ApexGO uses Bayesian optimization to choose where to explore next. An AI predictor estimates potency, results guide the following proposal, and constraints keep the search near a useful template. The computer is not proving a peptide works. It is prioritizing which sequence deserves the next real experiment. The team began with 10 peptide templates mined from extinct organisms. For each template, ApexGO proposed optimized derivatives. Researchers selected 10 per template, chemically synthesized 100 peptides, and tested them against 11 clinically relevant bacterial pathogens, including strains resistant to conventional antibiotics. 86 of the 100 synthesized peptides show detectable antimicrobial activity against at least one tested strain. Across the overall comparison reported in the NIH summary, 68% performed better than their parent template. When the paper focused on gram-negative pathogens, the improvement rate was 72%. Those numbers are meaningful because every point represents a physical compound tested in the laboratory, not merely a score on a computer screen. The approach also produced information that was less dramatic, but scientifically important. Predictions and measured results were related, but not perfectly. Biology still surprised the model. That is why the loop matters. AI narrows the search. Chemistry and microbiology decide whether the proposal survives contact with reality. The researchers then moved a small subset into 2 mouse models of infection caused by drug-resistant Acinetobacter baumannii. Some optimized peptides reduced infection more effectively than their original templates. In those preclinical models, selected candidates performed comparably with or better than a powerful last-resort antibiotic used as a benchmark. This is where we separate a promising lead from a medical breakthrough. Activity in a dish shows an effect under controlled conditions. A mouse model adds information from a living system. Neither proves a peptide is safe, stable, manufacturable, correctly dosed, or effective in people. The comparator is preclinical context, not therapeutic equality. Human trials have not happened, and no ApexGO peptide is an approved treatment. Peptide medicines face practical obstacles. Enzymes may break them down quickly. They may not reach the infected tissue at a useful concentration. A sequence that damages bacterial membranes might also harm human cells at the wrong dose. Manufacturing, storage, delivery, cost, and the possibility of bacteria developing resistance all remain part of the development path. The study's authors point directly to this next challenge. Future optimization must consider several properties together, including stability, pharmacokinetics, and safety. That is a harder objective than maximizing antibacterial potency alone. A useful drug is not the molecule that wins one metric. It is the molecule that clears many gates without failing a critical one. This is where AI may become most valuable, not as a replacement for scientists, but as a system for navigating trade-offs. A model can explore more sequence variations than a human team could enumerate manually. It can learn from experimental feedback and suggest the next informative candidates. Researchers can then spend laboratory resources on a smaller, better justified set. The same idea extends beyond peptides. Drug development is full of enormous search spaces, expensive experiments, and competing objectives. Generative systems can propose. Predictive systems can rank. Automated laboratories may eventually test cycles faster. But each layer needs transparent data, independent validation, and safeguards against models optimizing the wrong target. There are also questions of generalization. ApexGO was demonstrated with 10 templates and specific pathogen panels. A model that performs well here may behave differently with unrelated peptide families, new bacterial species, or resistance mechanisms it has not seen. Repeating the work across laboratories and broader biological settings will tell us whether the approach is robust or unusually suited to this experiment. For patients and clinicians today, the result does not change which antibiotic should be used. It does not justify buying an unapproved peptide, avoiding prescribed treatment, or assuming AI has solved antimicrobial resistance. Its value is upstream, a more efficient way to turn molecular possibilities into candidates worthy of serious testing. AI did not invent a cure for antibiotic resistance. It helped redesign peptide candidates, and many improved on their templates. That could focus early antibiotic discovery. Next steps remain demanding: validation, safety, manufacturing, dosing, and human trials. ApexGO is a research tool and a reminder that strong medical AI connects computation to careful experiments instead of replacing them. AI can search molecular possibility at a scale no research team could explore by hand. The achievement is not skipping the scientific process. It is choosing better experiments within it. If that advantage holds across more pathogens and more stages of development, it could help replenish an antibiotic pipeline that urgently needs new ideas. This video is general research education, not medical advice. If you are prescribed an antibiotic, use it exactly as directed and discuss questions with a qualified healthcare professional.